Are you taking AP Biology? Then you are in the right place, because for the next hour or so, I'm going to be going over Big Idea 4 to help you get ready for the AP Bio exam that's happening on May 10th at noon, in case you didn't know what time it was at. So I'm Tiffany Jones, and I'm coming to you from AP Bio Penguins. So you may be wondering, well, why are we calling us penguins? So we're calling ourselves penguins because of the fact that you, as an AP Bio student, are dressed for success. And so now that you're in this live and you're sitting here with me, then you are now a penguin. Yay! So real quick, let's go on. Okay, so today what we're doing, is we're doing Big Idea 4. So if you've been in any of the other sessions, you know that there's like a bunch of topics that kind of all correspond. So I'll go through all the topics that correspond with Big Idea 4. So the different topics we have, we'll talk about water, we'll talk about the macromolecules, we'll talk about the cell and the different structure of and function of the different organelles. We'll talk a little bit about energetics and specifically looking at fitness within the energetics. We'll look at genetics, specifically how we can make different genotypes and how those genotypes can allow for different effects in evolution. And then we'll end it with a little bit of population ecology, talking about the interactions of organisms and how that affects the number of organisms we have in an area and how diverse, and whether we can bounce back from some type of disaster that might take place. So without further ado, and I've been running over an hour. So again, if you need to log off, this is saved. This is always going to be a Market Learning page. And then if you do need to find it, you can always go to my website, apbiopenguins.weebly.com, and you can find basically all of my resources on there. So 1.1 is on water. So we need to think about how water and its polarity works with hydrogen bonding. And so we need to think about what is the structure of water. Okay, well, water is polar. And water is polar because of the fact that it has polar covalent bonds. So if you remember back to chemistry, a covalent bond means that it's sharing valence electrons. And so a polar covalent means that there's an unequal sharing. So the oxygen is more electronegative than the hydrogen, which means that it kind of pulls the electron kind of closer to it, making the oxygen partially negative and the hydrogen partially positive. And so due to that, we get a hydrogen bond. So the hydrogen bond is an attractive bond between water molecules. So a lot of students struggle with that. They kind of think that the hydrogen bond is actually in the water. It's not. It's between the water molecules, or it's between the water molecule and something else, or between, actually technically, an electronegative atom that's bonded to a hydrogen that's attracted to another electronegative atom. So you can see here that water has a lot of these different hydrogen bonding in it. And because of the hydrogen bonding, it gives a lot of different features and kind of properties to water. So first thing is cohesion. Cohesion is where water is going to be attracted to other water molecules. So here you can kind of see these two water molecules, they're kind of holding to each other. That's what's giving us a lot of the different kind of like beads on like if you were to wax your car and you see all these little beads of water, that's because they're kind of all coheting together. That's why the water molecules will come up with the capillary tube because they're all attracted to one another. The second thing is adhesion, water being attracted to other polar substances. So here you can kind of see that the water molecule is attracted kind of like the sides of this tube, okay? And the same thing happens when you look at a meniscus. So if you're looking at a graduated cylinder, you know how it kind of climbs the sides? That's because of the fact that glass is polar. It's, oh my God, my mind's currently going blank. Anyway, so the water is attracted to the, I think it's like silicon dioxide. Anyways, so it's attracted to the walls of the glass, and so it kind of climbs it. And so then you can see that together we get capillary action. Capillary action is the cohesion and the adhesion. So the cohesion is all of the water molecules attracted to one another, and the adhesion has to do with them going to the sides of the xylem, which is where in the plant it kind of holds to. And so what we see here is that, in the leaves, we're going to have evaporation. Water is going to be used in photosynthesis. We're going to see that the water gets evaporated, which will pull up some water from the top. Well, because you pull from the top, all those water molecules are attached to each other, and so you end up pulling from the roots. And that's how the water gets from the roots all the way up to the leaves, is because they're all attached to each other, as well as the adhesion of them sticking to the side, not sticking, attracted to the sides of the xylem, or the vessel in a plant. So surface tension is another component that's important with our properties. So due to the cohesion, there can be like a surface that you have on water. This allows you to skip rocks. And so when the water, like when the rock is skipping on the water, it's not breaking that surface tension. So it's because each water molecule has 4 different hydrogen bonds to other water molecules. And that allows to create that like tension that some organisms like a water strider can actually walk on the surface of the water. Because of those hydrogen bonds, water is also less dense when it's solid. Think about when you go have that ice cold water, ice cold iced tea, or whatever that you put ice in. The ice always goes to the top. Why does it go to the top? Because ice is less dense than liquid water. And so those hydrogen bonds kind of cause there to be like space in between these water molecules when they solidify to make the ice. And so that just creates like just empty space. And so it takes up more volume, which causes it to be, of course, lower in density. So this is good for a lot of organisms. If you think about living up north, which I live in Georgia, so this doesn't happen here, but up north, their lakes freeze over every winter. And it's because of the fact that, you know, it's less dense that the entire lake doesn't freeze. So those organisms that live in the water are able to survive the winter because the ice acts as a temperature buffer. It keeps that water still liquid. It allows for those organisms to continue to survive through the winter. So yay for water being less dense. It's also a universal solvent. You can dissolve any ionic substance as well as any polar substance into water. It's not going to dissolve non-polar substance, but it will create that solvation kind of, it'll surround it to kind of help to create these little vacuums of them together. And then last thing we need to talk about is hydrogen bonding that we'll see in our macromolecules. So when we look at proteins, you're going to see hydrogen bonding taking place in the secondary structure of a protein. So if you remember, the protein, which we're talking about in a second, is made of amino acids, and there's going to be the amine group and the carboxyl group. So between the amine group and a carboxyl that's upstream, there's going to be a hydrogen bond, and that creates that alpha helix or the beta pleated sheath that we see in proteins. It also can work in our secondary and our quaternary structure, which is where the R group is interacting. So if there's hydrogen bonding in the R groups of different amino acids, that can create that 3-dimensional structure that we'll see in proteins. Nucleic acids, the hydrogen bonds are important because it's going to be between the nitrogenous bases. So DNA is going to have 2 strands, right? And so the hydrogen bond will take place between the nitrogenous bases, and that creates your double helix. And we'll talk about that a little bit more, but you saw that when we looked at replication, how you would have to break those hydrogen bonds. Remembering helicase broke those hydrogen bonds so that we could then pull apart the 2 strands. So that is our quick review of water. Moving into biological molecules. Now, we're going to kind of cycle back a couple times with this. Like, I try to break them up by the topic. So there's some stuff that's in one topic, but not in another. And it's just kind of weird. So I'm going to do my best to try to like not give too much in each point. So first thing is on how do we make macromolecules, or how do we break down macromolecules? So hydrolysis is where we're going to break down a polymer. Okay, so a polymer is just a long string of monomers. And so hydrolysis, break it down by the words. Hydro meaning water, lysis means to break. So I'm going to break a water molecule. And when I break that water molecule, I'm going to break a substance. So here I see maltose. Maltose is just a disaccharide. It's made up of 2 sugar molecules, these monomers. And so we're going to add water. And by adding water, I'm going to be able to break this bond right here. As you see, the hydroxide or hydroxyl group is going to go to one side, while the hydrogen goes to the other. And so that creates, you know, the 2 molecules of glucose. I can break this polymer down into its monomers. And the opposite we have is dehydration. So dehydration is also known as a condensation reaction. So when I ask my students, I was like, hey, you know, what happens when you're dehydrated? Well, they're like, oh, well, we're missing our water. So in a dehydration reaction, we're going to remove water. So as you see here, I've got the hydroxyl group, I've got the hydrogen. So these 2 side groups are going to come off. That will make our water, and then we're going to create that bond there from, of course, you know, when you broke the OH off and you broke the H off, now those things need to make their other bond. So it creates the bond between them. So this is how we can do dehydration and form polymers from monomers. So there are 4 different macromolecules. So we're going to work through each of those macromolecules. So the first one we want to talk about is nucleic acids. Nucleic acids are made up of the nucleotide, a monomer called a nucleotide. And the bond that you'll see between it is a phosphodiester linkage. And this will be between the phosphate of one group and the hydroxyl of the other. And we'll talk in a little bit, but the phosphate is your 5' end and your hydroxyl is your 3' end. That all has to do with the counting of the carbons. But so when we're looking at it, we're looking at that there's a bond forming between the phosphate of one and the hydroxyl of the other, and it's called a phosphodiester linkage. So this is what the nucleotide looks like. You're gonna have a pentose sugar. So it's either gonna be deoxyribose or ribose. When I say pentose sugar, I just mean that it has 5 carbons to it. So there's like oxygen here. So it's like 1, 2, 3, 4. And then there's like a 5th carbon here that comes out to bond. And so deoxyribose is going to be found in DNA, and ribose is going to be found in, of course, RNA. There's going to be a nitrogenous base. So that nitrogenous base can either be adenine, thymine, cytosine, guanine, or uracil. Remembering that thymine is only found in DNA and uracil is only found in RNA, but the other 3 are found in both. And then they all have that phosphate group. So this is kind of the structure that we're going to see repeating when we look at a nucleic acid. And so in a protein, the monomer is an amino acid. The bond between it is a peptide bond. And so that's going to be between the carboxyl group of one and the amine group of another. And so between those two is where we'll see that peptide bond. So sometimes students can remember polypeptide because you have multiple peptide bonds. So it's all depending on how you remember it. And there's going to be a C terminus and N terminus based on whether we have the carboxyl group or the amine group on which side of it. So here you can see that monomer or the amino acid, it's going to have the amine group. So the NH2, it'll have the carboxyl group, the COOH. It'll have always a hydrogen. And this R group right here is variable. It can differ, it can change depending on which amino acid we're looking at. Okay, so it's important for us to understand that these can be different things. So they can either be hydrophilic. So if the R group is hydrophilic, it means that it's some type of polar. So it's probably going to have a lot of oxygens. They'll have sulfur in it. There'll be nitrogen in it. That's going to have these polar covalent bonds in it. And so it's going to be hydrophilic or water loving. And so when the final protein folds, it's going to fold to the hydrophilic. Our groups are going to face toward the exterior. It's going to fold because our world is water, it's aqueous. And so, of course, since it's water loving, it'll face toward that water. It could be a hydrophobic R group. If it's hydrophobic, that means it has a whole bunch of non-polar. So you have a benzene ring, you've got a whole bunch of carbon hydrogen bonds. There's not really an oxygen, sulfur, nitrogen in that R group. And then charged is usually going to be whether it's acidic or basic. It'll have a positive charge, or it'll be negatively charged because it's in its ionic format. And so that's also going to fold toward the exterior. Now, if you have questions, you're welcome to throw them in the chat, by the way. Like I forgot to mention that at the beginning, but you can throw those in the chat and I'll answer them as I see them. There's like a little lag between you and me, but I'll answer them as soon as I can. And so when we look at the polypeptide or the long string of these amino acids, we're going to have an N terminus or the side where the amine group is, and we're going to have a C terminus, the side where the carboxyl group is, because you'll have this long string. And that peptide bond will be between the carboxyl group of one and the amine group of the next. So carbohydrates, the monomer is going to be a monosaccharide. The bond between is a glycosidic linkage. Here you can see a glucose, which is just a monosaccharide. And so this is one of the ones that you might see often, C6H12O6. You're going to notice that carbohydrates have a lot of carbons, hydrogens, and oxygens, versus when we look here at, the nucleic acids and the proteins, they've got nitrogen, there's sulfur found in the proteins, there's phosphate found in the nucleic acids. But in terms of the carbohydrate, all it is carbon, hydrogens, and oxygens, and it's usually in a 1 to 2 to 1 ratio. So it's important that we understand that this usually makes these big polymers, right? So there's 4 different ones that are important to know. 2 of them are structural, 2 of them are storage. So cellulose, we're going to see in the cell wall of plants. Chitin you'll see in the cell wall of fungi, as well as it's making up the exoskeleton. So when you step on a cockroach and you hear that crunch noise, that's the chitin that you hear. We've also got storage. So like our starch that we found in plants, and then our glycogen is going to be found in animals. So you store your glucose in the form of glycogen in your liver, while plants are going to store their glucose in the form of starch. So if you eat a potato, think about how the potato is just the storage for the plant. That's why they call it a starchy food. And so when we think about this, it's important to understand that these bonds are slightly different in these polymers. So if you look here at starch, it's got a 1,4 alpha linkage. Okay, so that means that these side groups, these CH2OHs, are on the same side, is really what it comes down to. Versus our cellulose, they're going to be a beta linkage. So they're going to kind of, the structure flips over and over. Okay, now you don't have to know that, but you do need to understand that because of this different structure, we're not able to break down cellulose. So we as animals cannot break down that beta linkage. We can only break down that starch or the alpha linkage. And so there's a symbiotic relationship between organisms, microorganisms, that will break that bond for us. And so you think about like termites, right? So a termite eats wood, logically. So it needs to be able to break down cellulose. The microorganism breaks down the cellulose, providing the glucose to the termite, and the termite provides, of course, the food to the microorganism to help break it down. And so they have created that mutualistic relationship. So if we were to go and, I don't know, radiate the termites and get rid of their microorganisms, the termites are going to starve because they can't break down that food. They're obligate mutualism. So our last macromolecule we have are lipids. There is no monomer for them because they're not considered polymers. They're macromolecules. They're big structures, but they're not actually they're not like polymers. And so the big thing that keeps all the lipids together is that they're all nonpolar. Every single one of them is nonpolar. And so the first one we have is fats. So fats are made up of 3 fatty acids plus a glycerol. So the glycerol is right here. It's an alcohol. You see it has all these these oxygens here. This makes an ester linkage. I don't really think you need to know that one, but in case you were wondering, it's an ester linkage. And then you've got these fatty acids that come out. And fatty acids are just these big long chains of carbons and hydrogens. Now there's 2 different types of fatty acids we can have. There's saturated fatty acids and there's unsaturated fatty acids. Saturated fatty acids means that every single carbon is completely saturated with hydrogen. So if you see here, all these carbons have as many hydrogens as possible. So the interior ones all have 2 hydrogens and this exterior one has 3. Versus an unsaturated fatty acid is going to have at least 1 double bond. So if you look here, you see that there's a double bond in the structure. And that double bond causes there to not be 2 hydrogens on this carbon. So every single carbon is not fully saturated. So we call it unsaturated. Notice here that it creates this kink. So because of that kink, these two fatty acids can't bond together really closely. And that'll be important when we get into our phospholipids. But in terms of like fats in general, if you're wondering, like if they can compress, that's why they're solid at room temperature. If they can't compress, that's why they're an oil at room temperature. So if you think about like your, olive oil versus your coconut oil, my husband went and bought coconut oil the other day or a couple months ago, put in the cabinet and it was solid. And then the house got so warm that I opened up the cabinet and there was just this liquid in there. And that was because of the fact that it's a lot of these more saturated than unsaturated. So the other one we have is phospholipids. Phospholipids are made up of a phosphate group and then 2 fatty acids, but it still has that glycerol. So instead of there being this fatty acid right here, we just put a phosphate group there. So that's the only big difference between the fats and phospholipids. But because the phosphate is negatively charged, so it makes it hydrophilic or water loving. And so these are going to associate themselves to make our cell membrane. So we'll see the phosphate heads on the exterior of the membrane, and then you'll see your fatty acid tails on the interior. Hydrophobic tails, hydrophilic heads, phosphate heads, fatty acid tails, however it is that you want to remember it. That's why you make a bilayer for your phospholipid. Bilayer is because of the fact that there's 2 and they face one another at the interior. That's why the interior is nonpolar and you can't get things to move across it. It has to be small and nonpolar to move across, or if it's polar, it needs to have a transport protein. And also we have our steroids. Steroids are going to be made up of 4 fused rings. As you see, there's all these like carbon rings, which makes them very nonpolar. And then we've seen this before with ligands. They're intracellular signals. So like estrogen, testosterone, all of those can pass directly through that plasma membrane. And the receptor is going to be intracellular or on the inside of the membrane. And that's all. Okay. So the directionality of these molecules is essential to know. Okay, so nucleic acids are going to have a 5' end and a 3' end. As we mentioned before, it's because it's on the 5th carbon. So 1, 2, 3, 4, 5 is right here on this 5th carbon right here. And so that's why we call this a 5' end, is because it's on that 5th carbon. Versus over here, when we look at it, 1, 2, 3, the hydroxyl group is on my 3rd carbon. And so that's why we call this a 3' end. And so we're always going to talk about 5' to 3'. That's essential and very, very, very important. And so when we talk about replication, DNA is going to be read 3 to 5, or it's read in this way, but it's going to synthesize 5 to 3 because DNA is anti-parallel. Same thing when you talk about transcription. You're going to read the DNA 3 to 5, and you'll assemble the RNA 5 to 3. They're anti-parallel to each other as always. So there's important part is in the base pairing of it. So you're going to see that adenine is going to base pair with thymine, and there's going to be 2 hydrogen bonds. The way I remember that is it requires 2 lines in order to make a T. And then a cytosine is going to bond with guanine to have 3 hydrogen bonds. When I remember it's 3 is because the C is the 3rd letter of the alphabet, so I can remember that there are 3 hydrogen bonds. The way that I remember what bonds together, you can say at Conyers, Georgia, somebody talked about like that there are apples in the tree and cars in the garage. So that might help you to remember. It's all depending on what you need. And as I said before, there is a directionality. You're always 5' to 3', and they're anti-parallel. So they're going to be equidistant apart, but moving in opposite directions. So our proteins, as we said before, you've got your 2 amino acids, and they're going to bond together to create that peptide bond. As I said, there is a N terminus and a C terminus. That's the directionality that we need to make sure we know. And so we also need to talk about the structure that we have with this. So the primary structure is just that string of amino acids. All the amino acids bonded to one another. And as we said before, the bond is a peptide bond. Okay. Your secondary structure is your alpha helix or your beta pleated sheet. And so this has to do with the hydrogen bonding with the backbone. So the amine group of one amino acid and then the carboxyl group of another way upstream, when it folds back, it creates that hydrogen bond. And that's what creates the alpha helix or that beta pleated sheet. Tertiary structure is any bond that you've ever talked about in chemistry. So talk about covalent bonds, ionic bonds, hydrogen bonds, van der Waals forces, hydrophobic interaction, disulfide linkage. All of those are bonds that can take place because we're looking at the R group is binding to another R group of another amino acid. And that's what creates that final three-dimensional structure. So when we talk about one polypeptide, it only gets up to that tertiary structure. But if we're talking about like hemoglobin, then that's when we have multiple polypeptides, and those can form into that quaternary structure. And so that's the R groups of different polypeptides interacting that creates that quaternary structure that we see. So let's talk about stuff. I'm running way behind schedule. So the nucleus is made up of, it has this double membrane, and its job is that it is going to store the genetic material. It's going to be in charge for synthesis of our RNA. This is where we're going to see the ribosome assembled in there before it moves out into the cytosol. The rough ER is going to be a membrane. It's studded with all these little ribosomes on it. That's why we talk about that the ribosomes that are on the ER, it's going to be allowing for protein synthesis for secretion, protein synthesis for membrane proteins. It's also used for compartmentalization to kind of break up the cell. That's why eukaryotic cells are able to be so big. It's because they have compartmentalization with all these different organelles we're about to talk about. It also provides mechanical support, and then it has a role in intracellular transport. So it allows for materials to move through the cell due to this rough ER. So the smooth ER is connected to the rough ER. It just doesn't have ribosomes on it. It's folded in the same way that we might see the cisterna of the Golgi. So it has all these little tubes that are all structured. And so its job is for detoxification. It's also going to be in charge for calcium storage. So if you talk about muscle contraction, it's the sarcoplasmic reticulum, which is a smooth ER that's going to hold all the calcium to release it out as a secondary messenger. We also see that it's in charge of lipid synthesis. So this is where we're going to synthesize our protein hormones. The Golgi complex is going to be like this bunch of folds of little membrane, and that makes the Golgi. And so things move from the cis side to the trans side. So cis is going to be on the same side as the rough ER, and the trans is going to be on the plasma membrane side. And so during the time and the protein is moving through the Golgi, it is getting modified, it is getting repackaged. It's just, as they said in middle school, it's the mailman, right? And then the ribosomes are made up of a couple different things. You're made up of rRNA as well as protein. There's going to be a large subunit and a small subunit that we talked about when we looked at translation. The large subunit is going to bind to the tRNA. The small subunit is going to bind to the mRNA. And that's going to aid in the process of translation or synthesizing our polypeptides. There are two different types. There's bound ribosomes and there's free ribosomes. Bound ribosomes are going to be attached to the rough ER membrane. Free ribosomes are going to be ones that are just hanging out in the cytosol. Now, it's important to understand that all ribosomes start out being free ribosomes. Once they get a signal, they then move to the membrane to continue their process of translation at the membrane. But they do all start out being free. And function as protein synthesis. So we also have mitochondria. Mitochondria has a double membrane. There's that outer membrane that's smooth. The inner membrane is highly folded. That is going to be essential when you think about cell respiration. And so the functions of it is a set of oxidative phosphorylation, specifically taking place that inner membrane or this cristae. And also on the inside in this matrix area where we're going to see the Krebs cycle. And so in case you forgot or didn't realize it, yes, mitochondria has its own DNA as well as it has its own ribosomes. It is not part of the endomembrane system. And it's also believed to be a prokaryote that's in an endosymbiont or it has a symbiotic relationship that's inside of our cells. So same thing with our chloroplast. Our chloroplast is going to have a double outer membrane, and then there's going to be these little stacked thylakoid stacks, which are going to be called grana. And so this is where photosynthesis takes place. In the membrane of the thylakoid is where we're going to see our light reactions. And then in the stroma is where we're going to see the Calvin cycle. And so this is where, of course, photosynthesis takes place. And yes, they also have their own DNA and they also have their own ribosomes because they're also endosymbionts. So lysosome is responsible for intracellular digestion. So when you go through phagocytosis, I'll answer your question in a second. When you go through phagocytosis and you engulf food molecules, you're going to see that the food vacuole is then going to fuse with the lysosome for that digestion. It has these hydrolytic enzymes that are responsible for hydrolysis or breaking down polymers. And so it's going to break down those materials back into the fundamentals that are needed for the cell so they can recycle different organelles or different materials. It's also responsible for programmed cell death or apoptosis. And so there's that. And then our vacuole, there's multiple different types of vacuoles. There's a food vacuole that I mentioned before. So when you go through phagocytosis and the cell engulfs something. So I'm talking about the immune system with my students right now. So we talk about macrophages, how those macrophages are going to engulf bacteria that's invaded your body. So when it does that, it forms a food vacuole around that bacteria, and then that food vacuole fuses with the lysosome for digestion. There's also going to be a central vacuole that's found in plants. This is responsible for water retention that creates that turgor pressure. So we know that plants like to be in a hypotonic environment, all that water rushing in. And because of the central vacuole, it can hold that water to create that pressure on the wall, which provides the rigidity that we need for our plant cells. And then we also have a contractile vacuole, which is going to be found in some of our kind of organisms that live in hypotonic environments. As the water rushes in, contractile vacuole is going to keep pumping. And it's going to push out the excess water so that those cells don't burst. So there is a question in the chat. It says, so the ribosomes only stick to the rough ER when it has to make a protein? No. So the ribosome is decontached. So you have a small subunit and a large subunit just chilling out on the cytosol. So the small subunit is going to bind to the mRNA. And once it gets to the AUG, the start codon, that's when the large subunit comes and binds to it. There must be some type of signal that we don't really need to know about. And those come together. So as it's translating, there's going to be a signal in it. It's the, oh gosh, I cannot remember the name of the signal, but there's a signal on it. And that signal causes it to move to the membrane, which causes it to bind at a specific receptor on the ER's membrane, and that is what allows for the finishing of the signal. I want to call it the SRP, the signal recognition protein, I think is what it is, like the SRP is where it's binding. So I think I answered your question. I hope I did. If not, just let me know. So 2.2 is again just talking about all the different organelles, and I mentioned almost all of this already, so I wanted to just include it so that it's in the PowerPoint, so when I export and you have a copy of this, you have it to review. So just a reminder, ER is mechanical support, protein synthesis on those membrane-bound organelles. It's important for our intracellular transport. Mitochondria is important that we know that there's a double membrane that compartmentalizes for the different metabolic reactions. As we said before, the Krebs cycle is where we're going to see, is going to be in the matrix of the mitochondria, and the electron transport chain and oxidative phosphorylation and chemiosmosis takes place at the cristae within the mitochondria. Lysosomes are going to be important because they, create that compartment for digestion with the hydrolytic enzymes. They'll recycle the organic materials for the cell, as well as they're good for apoptosis. And then, of course, the vacuoles are going to be for storage and the release of macromolecules and different waste products, as well as they can retain water for turgor pressure that we mentioned earlier. It's also important that we understand like the folding and how the folding creates for different compartments in the mitochondria and our chloroplast. So the folding of the mitochondria, that cristae, creates a larger surface area. That larger surface area provides more spaces for more electron transport chain and then more chemiosmosis to increase the amount of ATP that is made in the mitochondria. The chloroplast, of course, is the thylakoid and the stroma. Thylakoid is going to be the site where our light reactions take place. So it's important that we understand that is also increasing our surface area. And then the stroma is where we're going to see the Calvin cycle taking place for photosynthesis. And then they literally break down all of them and make sure that you know all the parts of it. So light-dependent reactions and photosynthesis will be in the grana. The carbon fixation or the Calvin cycle is going to also take place in photosynthesis in the stroma. The Krebs cycle or the citric acid cycle, yes, you should know both of those words interchangeably because they have been used both on the AP exam. Is in cellular respiration, and it's in the matrix, which is on the inner part of the mitochondria. And then the electron transport chain ATP synthesis, I wrote cell respiration there because of the way that the answer was inner mitochondrial membrane. But don't forget there is an electron transport chain on the thylakoid. So you do see the electron transport chain in both of those. You do synthesize ATP for both of them. ATP for cellular energy is mitochondria. ATP for photosynthesis is in the chloroplast. So kind of make sure you do recognize the two differences there. So fitness, okay. Fitness, when you talked about it in evolution, you know that individuals that are more likely to survive are more likely to leave more offspring. And so we're going to see that they're going to have increased fitness because fitness just has to do with your contribution to the gene pool, how often, you're passing on those genes and whether your offspring are able to survive to also reproduce. So there's variation at the molecular level. And so because of this variation, it allows for the organisms to respond to environmental stimuli. Okay. So here we can see hemoglobin, and I'm going to be completely honest with you. I had no clue what the standard was talking about. So I actually did watch the AP classroom video, and these are the two examples that they use. So that's why I'm bringing them to you because that's the only thing I could come up with. So hemoglobin, when you're still in utero, you've got fetal hemoglobin. The job is so that it's a higher affinity to steal the oxygen from your mother's hemoglobin. And so here you can see that it has, a higher percent oxygen saturation at a lower partial pressure because it kind of has a higher affinity for that oxygen. And so this kind of shows you that if they're able to obtain more oxygen, then they're going to have an increased fitness. So different molecules can allow for you to have increased fitness. So there's that variability. And then variation in the number and the types of molecules provides a greater ability to survive and reproduce. So this one was talking about chlorophyll. And so you know that inside your chlorophyll. So I'm going to answer your question in a second. You've got chlorophyll A, chlorophyll B, and you've got the carotenoids. So chlorophyll A is going to kind of absorb light at different wavelengths than the B, as well as different wavelengths than the carotenoids. And so since you have multiple different proteins, multiple different pigments, they weren't proteins, sorry, they're pigments, different pigments within your chloroplast, then you're going to be able to absorb a wider range of light. And if you can absorb more light, you have more light energy to fuel photosynthesis. So this increases the fitness of the organism. As you notice, the only wavelength that it doesn't absorb is the green wavelength. And we all know that the reason why it does not absorb green is because it reflects it, because we see that it's green. Yay. So the question in the chat says, is it possible if Marco Learning can organize these videos by unit rather than big idea? Yes, they're already made by unit. I made them for the units last year. I just decided this year I would do by big idea to give you a different approach to the content. But yes, every single one of these is already saved on Marco Learning. If you go to their site, you go find their the video folder for AP Bio, you will find every video I've done for the past 4 years from them. There's one for every single unit. There's one now on all the big ideas. There's one on me going over math applications. There's one where I just did, quick overview of the whole unit, like all the year. There's tons of videos on there. So please go check out all those videos. If you want them organized differently, they're also organized on my website too. So environmental effects on the phenotype. So based on the environment, we might see different effects in the phenotype. We're going to see the organism look differently. Yes, these videos get loaded. They're going to automatically be saved to Marco Learning. So due to the environment, we're going to see different phenotypes. We'll see different responses of that organism. Okay, so here you can see this this rabbit, and so they usually only have like the dark black color fur on their ears and their nose and their feet, and I think on their tail maybe. And so they were like, okay, well, what would happen if it moved to other areas? So these areas of the body are usually colder areas, and so they found that the reason why they were dark is because of the fact that those are colder areas of the bunny, okay, or the rabbit. And so they said, okay, if I take the skin off of this part, and they take the skin off just so that you can get a closer connection of the coldness to the skin. If we take off that skin and we put an ice pack on there, what will happen to the color of the fur that comes back? And the color of the fur came back dark. And that was because of the cold, the environmental condition caused the phenotype to be different. So it's responding to its environment, even though it is the exact same genotype, even though its genes say make it white, because of the fact that the environment was cold, that caused the signal to change the phenotype of this. This is called phenotypic plasticity. So we also see here that the birds, if they lack a diet in carotenoids, they'll have a lot of little color. And so based on the type of food that they're eating, you're going to see differences in the bird species. And then the one that you all probably know about is hydrangeas. So these hydrangeas all have the exact same genotype. So their genotype says to be whatever color they are. And so based on the acidity of the environment or based on the aluminum content in the environment, they're going to be, between blue to pink. So even though they have the same genotype, completely different phenotype, that is phenomenal. It happens in the snowshoe hare. It happens in all types of organisms. I was watching a TikTok the other day. It happens in like these frogs based on predators or no predators in the environment. So all of this is constantly happening. And I haven't seen a question like this on the exam, but I know it's going to be coming soon. And I also haven't seen a multiple choice. They haven't released those to us. So there could be questions in the multiple choice that I just haven't seen. So chromosomal inheritance, this brings us back to meiosis for a second. I don't know why it's here, but it's in the standard, whatever. So first thing we talk about is crossing over. So during prophase of meiosis one, prophase one, you're going to see crossing over. So this, somebody was asking what is a homologous chromosome before. This is a homologous chromosome. So it's the chromosome that is of the same length, it's the same chromosome, but the maternal and the paternal chromosome. And so these non-sister chromatids are going to overlap and they'll exchange genetic information. So what I've done before, and this may or may not work, but we're gonna try it. And so here's our paternal, here's our maternal chromosomes. So together they make that tetrad, okay? And so what happens is that they're going to literally exchange genetic information. So they cross over, and when they cross over, now you can end up with 4 different kind of products that come out of it. One that's completely paternal, one that's completely maternal, mostly paternal, maternal with a little bit paternal, and mostly paternal with a little bit maternal. And I think I messed up with my hands. I'm sorry, y'all. I was just trying to do that fast. Anyways, so that gives you this different combination that we see. So we also have the independent assortment. Independent assortment has to do with that the homologous chromosomes can independently align on that metaphase plate. And so based on how they line up, so here, let's assume there's only 2 chromosomes. They can either line up where all the maternals on one side and all the paternals on the other, or they can flip and flop. And so the different number of options you have is 2 to the number of chromosomes. So since we have 23 different pairs of chromosomes, it's 2 to the 23rd, which I think is like 7.4 billion combinations or 8.4 billion. I can't remember the number. But it creates a whole lot of combinations. This only gives us 4 different options. And then the law of segregation has to do with during anaphase, your alleles are going to move apart from each other. So as you see here, the dominant allele moved apart from the recessive allele, this dominant allele moved apart from the recessive allele. So they can actually segregate into different cells. I'm sorry. And then random fertilization refers to that any sperm can fuse with any egg. And so this is how we can get genetic variation within our population, is because crossing over creates a new combination. Independent assortment makes new combinations within our gametes. Same thing as law of segregation that piggybacks on law of independent assortment. And then random fertilization, you've got the 7.4 billion combinations coming together. So it creates this huge, unique organism that is sitting right here on YouTube listening to me talk crazy. So this leads us into chromosomal base of inheritance. So organisms are inheriting chromosomes. And so the genes that you have on those chromosomes are the ones that you're inheriting. They're inherited from the parent to offspring. And somebody was asking questions in, I think, Reddit. They were asking about, well, if your grandparents had a trait, how does it respond to you? Well, you have your grandparents' alleles the way that your parents were made from one set of your grandma and one set of your grandpa that made your mom or your dad, whichever one. And then those crossed over during their prophase. So you actually have a combination of both your grandma and your grandpa's genes on your chromosomes, which is kind of cool and interesting. And then there's a lot of different genetic disorders that can take place. So up here, we're talking about kind of like a single affected mutated allele. So like sickle cell disease, which we know is going to help in malaria. It also causes, of course, the disease where if you have a low amount of oxygen in the environment, it causes your cells to go from that normal kind of concave shape to like the sickle crescent shape, which is going to cause a lot of pain. It can kind of lead to the blood kind of building up in different areas and clotting. It makes you lethargic or kind of like tired all the time because of the fact that you can't carry as much oxygen. So a lot of problems with that. Tay-Sachs disease is another example of this. This is, I believe, a neurological disease. They're not going to require you to know this. I was just giving you examples. And then Huntington's disease is a autosomal dominant trait that sometimes shows up in these pedigrees. Again, they're not going to tell you what the disease is because they're assuming that you're going to have a background to it. So you're going to have to just apply what you see in the diagrams or the figures for what they are. In terms of chromosomal changes, you can have Down syndrome, which is also trisomy 21. So remember, on March 21st, which I believe is in 3 days, okay, wear crazy socks because it's Down Syndrome Awareness Day. So you want to wear crazy socks. The sock is kind of like a chromosome, like it looks like a chromosome. So make sure that you do that and celebrate with them. Klinefelter is XXY, so it's an individual that has 2 X chromosomes and a Y chromosome, and then Turner syndrome is an X and an O, because of the fact that they only have one X chromosome, and then they don't have a second allosome. And yes, allosomes are now sex chromosomes, in case you were wondering what that was. And so here you can see a karyotype of an individual with Down syndrome. So you can see that they have 3 sets of chromosome 21. So this individual gained an entire extra chromosome. This is due to something called non-disjunction. So during anaphase 1 or anaphase 2 of meiosis, it didn't happen. And so the individual got 2 of chromosome 21 in that egg or that sperm. And so when that 2 fused with the 1 from the other gamete, it gave them 3 of chromosome 21. And this happens with a lot of other things like Edwards syndrome. I can't remember all the other ones, but there's a couple different ones that result from, I think it's trisomy 15, trisomy 18. So it's all these like different ones where you have extra set of a whole chromosome. So you get variation within your population. If there's low genetic variation, this is going to cause problems in terms of our bottleneck effect. Certain alleles can become fixed, in which that's the only allele we have in the population. And because that's the only allele we have in the population, then a natural disaster can wipe it out. And so here we see this female cheetah. It's going to mate with multiple males. And by mating with multiple males, it's going to increase the genetic variation because there's, the 7.2 trillion here, 7.2 trillion here. And it gives us a lot of variance in the actual, I'll answer your question in a second. It gives us a lot of variation in the actual offspring. Okay, versus if the female only mates with one male, you've got the exact same alleles all mixing together. So it doesn't really create a whole different variation in that population. And so that can be, cause them to be subjected to, bottleneck effect that causes them to, have low genetic variation, which is bad for a changing population. So what we want is the environment or the population to be genetically diverse. So here you can kind of see antibiotic resistant, right? So within our population of bacteria, which I know we don't want the antibiotic resistance, but I'm just saying like this is an example. So you've got this population of bacteria, and because of the fact that there is variance in it, there are some of them that are already, not susceptible to the antibiotic. They're already resistant to that antibiotic. And so when you give the antibiotics, you put the antibiotics into that individual that has this bacteria, it's going to kill all the bacteria that don't have that resistance. And so we're then going to see that the population of bacteria can survive through this change in the environment. So somebody asked in the chat, what is the difference between a chromosome and a chromatid? A chromosome is your DNA. It's wrapped around your histones. A chromatid is going to be a duplicated chromosome. So when we talk about chromatids, we're usually talking about sister chromatids. So G1, they're chromosomes. S, they're sister chromatids. G2, we're looking at sister chromatids. Prophase, they're sister chromatids. Metaphase, sister chromatids. Anaphase, chromosomes. Telophase, chromosomes. Cytokinesis, chromosomes. So we're looking at if there's a duplicate. If there's a duplicate, we call them chromatids. If there's no duplicate, then we call them chromosomes. There was an FRQ a couple years back that caused some issues with some students. So basically, interphase, S phase, all the way through when they separate in anaphase, that's all chromatids, and then the other starts chromosomes. So origin of life, these numbers are directly in the standards, which makes you wonder if there could be a question that asks these numbers. I truthfully don't know whether they're going to ask them or not, but Earth was formed 4.6 billion years ago. The environment was too hostile for life until 3.9 billion years ago. And the earliest fossil evidence we have is 3.5 billion years ago. Those numbers are directly in the CED, directly in the standards. So my brain goes, oh, that's important for students to know. So I don't know how to remember those, but remember them. So early life. So how did life come about on Earth? So they did this experiment. This was our Stanley Miller experiment. So he went and he took, primordial water. He put in the sea. He kind of heated up, similar to like how the sun would heat up water, causing it to kind of evaporate and creating that water vapor. And then he created the same atmosphere that was on earlier. So it's got the methanes, the ammonias, the hydrogen gas. There's no oxygen present. And then he shocked it with electrodes, which provided kind of like lightning would create energy in the environment. So he had this, I think it was an oxidizing environment. And so then he put it through a condenser, which simulates rain. And then he collected that sample. And so he was able to abiotically synthesize organic material. So without life, he was able to make organic compounds. And organic compounds we know are like the carbon, hydrogen kind of structures. And that's organic. You'll spend an entire year on that if you go through organic chemistry. Yay. And then inorganic chemistry is just all the other elements for a half, like literally one semester. It's great. Anyway, so there's a lot of different stuff in the standards. They're talking about that you could mention that some of the organic materials came on a meteorite that came to Earth, or we could just talk about that we had precursors that took place, okay? And then the last thing to know about this is that originally it was an RNA world. So the genome was made up of RNA. Why do we know that it was the first genetic material? Because RNA can make more RNA, as well as RNA can make proteins. RNA is acting as a ribozyme, so it can synthesize itself. It can catalyze its own reaction. And then the last thing we need to talk about is some ecology. So what is a population? Population is just a group of organisms that are the same species living in the same area. So, you at school, that is a population in that classroom. There's a population of students. You then could build up a little bit higher and talk about the community of school, where you've got your teachers, your administrators, the janitorial staff, the secretarial staff, whatever you want to mention. That would be the community. But like the actual students would be the population. So population growth is referring to kind of how the population grows over time. So if you have calculus, you know that this is the derivative. So dn over dt. All this is looking at is the change in our population size over the change in our time. It's going to be B minus D. This is the actual equation that's in your standards. In the CED, where B is your birth rate and D is your death rate. So the number that are born divided by the number that exists, the number that died divided by the number that exists. And so that allows us to see what the change is. So if you take, the number of born minus the number that died, you can say, oh, well, it increased, it decreased by this many in that certain amount of time. So there's two different types of growth. We can have exponential growth. Exponential growth has to do with that there is an unlimited supply of resources. There's nothing that will restrict the growth. They've got enough food. They've got enough space. There's no competition. Like they are living like Romans. I don't know. They're just amazing. Okay. They're going to see this exponential J-shaped curve. Okay. So the way that we do that calculation is it's going to be R times N. So R is our intrinsic growth rate. R is actually equal to lowercase b minus lowercase d. And that's just looking at your intrinsic birth rate and your intrinsic death rate. And so they talked about the elephant population in my textbook, and how there was this, exponential growth of the elephants at one point. So you kind of see this, this extreme growth. It was due to a lot of laws that were put in place to protect the elephants, because for a while, elephants were being hunted for their tusks, their ivory, all that fun stuff. And so example math problem, because of course we all struggle with math and we need to see a little bit of math problems. If there's a population that's 400 individuals with a growth rate or rate of increase of 0.5, how many individuals would occur after 2 generations? So some of you in calculus may know a better formula, but on the AP exam, you may not remember that better formula. So I'm going to use the one that's actually on your formula sheet and show you how you would use that formula. So we're going to sub in where we have our R times our N, and this gives us dn over dt, or the amount of change over time. So this tells me 200. So I'm going to say, okay, well, it started out being 400. I add in the change, which was 200. So now after the first generation, I have 600 individuals. Okay. So the population isn't going to increase by 200 the next time. I need to recalculate. So I'm going to say, okay, well, R times N, because my R is 0.5 still, but my N is now 600. So 0.5 times 600 gives me 300. So now I'm gonna say, okay, well, the 600 that I started with plus the 300 gives me 900. And so my population would be 900 individuals after 2 generations with a growth rate of 0.5. And so here you can kind of see a prettier picture, but basically it's the same. So the difference is that I just wrote R instead of R max. Your formula sheet just has R on it, but either one of these can be used. R max is just talking about like the maximum rate of increase, but it's just talking about R. So an effective density. So density is just how many individuals we have in a certain area. Okay, a more densely populated area means that there's more individuals in this little area. So number of individuals over area. If it's a less densely populated area, then that means that there's a lower amount of individuals. And so there's different factors. You have density dependent factors. These are factors that depend on the population, about the density in that area. So they're going to increase when the population increases. So competition, the more individuals you have in a certain area, the more competition is going to take place. Predation, you have more predators, there's going to be a rapid decrease in the population of the prey. The predators are going to run out of food. There's a problem when there's more predators in the area. Disease. Why do we get sick with the flu during the wintertime and not the summertime? The flu's always there. It's not just like coming around during the wintertime. It's always there, but we notice it more, and it has a higher transmission rate during the wintertime because we're all inside during the wintertime. So we're more densely populated. We're all more congested in the same area versus during the summertime, you're just out and about, you're spending time outside. You, I assume, are in getting some vitamin D growth, all that fun stuff. Okay, whatever. Density independent factors. This is something that means nothing based on the density, doesn't matter about the size. So like a natural disaster, a tornado doesn't really come because there's more people. It doesn't move toward the populous area because, oh, there's more people there. Like, it is a natural disaster. It doesn't really care based on density. Human activities, all that does not really depend on the density. And so here you can kind of see that a fire is going to reduce the amount of food that's available for this jackrabbit. That would be considered density independent. Okay, the fire did not specifically go to that tree because of the fact that there were more or less of the rabbits. Okay, but because there is a decrease in this rabbit, we're going to see that the kit fox is also going to decrease because its food, the density, decreased, and because it had less food, its population is also going to decrease. So that leads us into logistic growth, okay? And so this is where you bring in the carrying capacity. That's what that K stands for, is carrying capacity. It's the maximum individuals that can be supported in a certain area. So another math problem. I'm sorry, I forgot to tell you what the formula is. The formula is R times N times K minus N over K. Yes, this is given to you on the formula sheet. I don't know why I put 200 there. I think I made a mistake. So if the population has 400 individuals with a rate of increase of 0.5 and the carrying capacity is 800, how many individuals would you have after 2 generations? Okay, so if I go ahead and sub this in, so here's my R, 0.5, my N is 400, and then my K we said was 800 minus 400, which is my N, divided by my K, which is 800. It's a 100. So since I had 400 to start, the change in population is 100. I now have 500 individuals. Then you calculate a second time, and then you're going to find that now we can have an increase of 75, and then we only have 575 overall. So notice that as the population increases toward the carrying capacity, the rate of increase is going to decrease. Originally, there was 500, right? Because we increased by 100. And then because there was more individuals, I only increased by 75. So there was less rate of increase. There's less individuals that were born because we got closer to that carrying capacity. The biodiversity, okay, so how diverse is the whole population? How diverse is our community? Okay, the more diversity that we have, the more increase there's going to be in the population. As things change, we can survive any of these disasters or these things that might take place, whether it's a pathogen or whatnot. Okay, so there's 3 different things to deal with that. So you have genetic diversity, so there's a whole bunch of variability due to the, independent assortment, crossing over, random fertilization. You can have species diversity where like the actual multiple different types of species in the area that interact with one another. And then you have ecological diversity. So that's the biotic and abiotic factors in the environment. Okay, so this is where we bring in keystone species, your producers, and your essential abiotic and biotic factors. They all contribute to the diversity of that ecosystem. So what's a keystone? Keystone species is an organism that is disproportionate to their abundance effort. Okay, so they are very important for their environment. And so if they are removed, it's going to cause the entire ecosystem to collapse. And that was an FRQ question a couple of years ago. And literally the students just had to say, since the keystone species was removed, the ecosystem collapsed. It was as simple as that. So you need to make sure you understand that if keystone's removed, the entire ecosystem collapsed because they have such a important component of it. And then so an example of this would be like our sea otters. So our sea otter is going to be a keystone species. So the sea otter eats sea urchins. Okay. If we see a decrease in our sea otters, so something happens, we lose our sea otters. Okay. Maybe they get eaten up during a Marshmallow Madness, if you're following Marshmallow Madness. And so we lose our sea otters. Okay. Well, that means that the sea urchin population increases. And because the sea urchin population increases, they eat all the kelp. So we'll see a decrease in the kelp forest, and all the other organisms that are dependent on the kelp forest are also going to see this massive decrease. So somebody says, can I explain the difference between exponential and logistic? The difference is carrying capacity. So there's only a certain number of individuals that can be sustained in logistic exponential growth. And so like it's going to reach that carrying capacity and the population is going to level out. Versus when we look at exponential growth, it just keeps growing indefinitely up. So that's the big difference we see is exponential is just going to keep excessively growing up. Bigger and bigger and bigger, versus in our logistic, it is maintained at a specific population. Most things are in logistic, like they may start out being exponential, but at some point they're going to run out of resources. Like nothing can survive forever. And then disruptions to ecosystem. So things that can affect our ecosystem, right? So first thing, invasive species. So as we keep moving around the planet, we keep accidentally introducing organisms to areas that we don't mean to introduce them to, okay? And so because of the fact that we introduce this organism to a new area, it doesn't have any natural predators. It has an unlimited amount of resources. There's nothing to limit its growth. And so we see this exponential growth rate, which is detrimental to some of our ecosystems. Okay. So first thing, like the brown tree snake. I'm gonna be honest, I just went on Google and search, invasive species just to get you some different examples. So the brown tree snake, they accidentally brought it to Guam. There was no animal that hunted any of these snakes. So these snakes then, started eating a bunch of birds and rodents and other small animals that before weren't being consumed as excessively. And so it caused there to be an extinction of 9 different organisms in Guam. So invasive species are bad. Y'all probably know about kudzu. I don't know about y'all up north. Like I know us in Georgia, we have a bunch of kudzu. I'm assuming you have kudzu up in your area. Maybe you don't. I don't know. Zebra mussels are another example. Okay, so this one was talking about that there was no natural predators. So because there's no natural predators, they excessively grow, right? Maybe they can outcompete the natives. So the big head and the silver carp, they're common in the Missouri River. They feed on phytoplankton, which are these small little organisms that float around in the water. The native species called the fat paddlefish, they also feed on that plankton, but they're not able to get the plankton as fast as the big head and the silver carp. So we're going to see a decrease in the paddlefish so they can outcompete the native organisms, which is going to, of course, decrease the amount that we have present. Okay, humans, we are wonderful people to this earth. And so we introduce a whole lot of new diseases that can ruin some of these native species. We change the habitat constantly. Deforestation is causing it. As we keep having all these emissions, these carbon emissions into the atmosphere, we're changing temperatures, we're changing pHs of the rain. We're causing so many problems due to us. Okay, there's also geological and meteorological events that can affect the habitat. And so here's just like a big graphic, kind of shows a bunch of them. So you can see like the oil erosion from us, working in different areas. You see the pollution that's being added. We're modifying habitats. We're introducing new species of all these things that are happening because humans are just wonderful people. So I know that I'm at the 1 hour. As always, you're welcome to log off, but I do want to make sure that I go through a couple questions with you. I have, I think, 1 multiple choice and 2 free responses, just to kind of help us see applications of some of this stuff. So scientists found that an existing population of certain species of amphibians are small in number, and they lack their genetic diversity. And they're separated from each other by wide areas of dry land, which the following human actions is most likely to improve the long-term survival of amphibians. So we talked about that they need to have genetic diversity. The genetic diversity allows for bottleneck not to happen. It's allowing for there to be able to come over any type of issue. Like if there's a disease, it's not going to wipe the entire population out because there's a multiple different variants in terms of their DNA and their genes. Think about, malaria, right? Malaria it does wipe out a lot of people. It shouldn't wipe out, but it does. But the individuals that have sickle cell are resistant. They're not going to be affected by malaria. And so because of that genetic variation, they're able to survive. So how can I increase the variability? How can I increase the diversity within my population? So that's what they're wanting us to come up with, a way that we can increase genetic variation. Okay. So cloning the largest individual to counteract the effects of aggressive. How does cloning an individual help me to increase genetic diversity? It doesn't. Reducing my population size by 1/5 to decrease competition. Again, I'm looking at genetic diversity. If I decrease population, I'm going to have even less genetic diversity, which is a problem. Okay. Construct a dam and irrigation system to control flooding. What is that doing for my genetic diversity? Building a pond in the area of dry land to promote interbreeding between separate populations. So these populations are separated. If they can come in contact with each other, can they reproduce if they're the same species? Yes, they can now reproduce. And so this population may have one gene, this population may have another gene. And by allowing them to now mate together, now I can increase the genetic variation and the diversity within my population. Okay. That's what they're doing in a lot of these areas, like the cheetah that's currently going through bottleneck, is they keep introducing more organisms to the area, more of the same population, so that they can increase new genes. There's an FRQ question about snakes from like 5 years ago that deal with the same thing, where there's just too many, like we see a decrease in snake population. How can we increase it? And it all has to do with introducing new snakes to the area. So for your response, and then only part A, and it's really funny because this question was actually on Reddit earlier today. Stu was asking all kinds of questions about it. I don't know, whatever. We're only doing A though. So they investigated the mode of inheritance, doesn't really matter, don't really care about any of that intro. The disorder alters glucose metabolism. Describe the atoms and types of bonds in a glucose molecule. So you have to think, okay, well, what's glucose? Glucose is a carbohydrate. What makes up carbohydrates? Okay, well, those are carbons, hydrogens, and oxygen. That's what makes a carbohydrate, right? Carbohydrate, like think about it. There's a carb and a hydrate. Hydrate is oxygen and hydrogen, like carbohydrate. Okay, so we know that it's carbon, hydrogen, oxygen, and then we have to think about what kind of bond is it. Well, they're all covalent bonds. So you talk about that the atoms are carbon, hydrogen, oxygen, CH and O, and they're held together by covalent bonds. Okay. Now again, it says to describe. Can I write out carbon, hydrogen, oxygen covalent? Nope. Please write a complete sentences. Please show me that you can understand what is making up it. My students, when they did this question, they mess up all the time. You keep telling me hydrogen bonds. Remember, hydrogen bonds are going to be between things. They're not actually in the structure of it. Okay, so it would be between the glucose and water, there could be a hydrogen bond, but in the actual thing, because it says the bond in the glucose. Okay, so here we have a question where we're looking at cystic fibrosis, genetic condition associated with defects of CFTR protein. It's a gated ion channel that requires ATP binding in order to allow chloride ions to diffuse across the membrane. Yes, I know we didn't talk about diffusion in this, but bear with me for a second. So in the provided model of a cell, draw arrows to describe the pathway for production of normal CFTR protein from gene expression to final cellular location. Okay, so gene expression, the DNA is going to be found in the nucleus, and that's where transcription takes place. So we will transcribe the gene, we'll make the mRNA. So we'll go from the nucleus, where? Well, we have to go to a ribosome. So you can come out here to this ribosome, and then you'll come to the rough ER. And then after we're in the rough ER, we're then going to go to the Golgi to modify it, and then we'll go to the plasma membrane. And so here's our nucleus. then we go to our Golgi, and then we go to the plasma membrane. Okay. And so I graded this one on the AP exam. You had to make sure that you stopped at that membrane. If you went past the membrane, you didn't get points because of the fact that you didn't demonstrate that you understood that it was going to be in the membrane. So be very careful that you're reading your prompts, because it tells you that it's allowing something to diffuse across the membrane, so it has to be found in the membrane. A lot of students went to the mitochondria, they got confused by the things talking about ATP binding. There's going to be distractors in there, okay? So you need to make sure that you ignore your distractors, that you just kind of say, okay, I don't have to use everything in this diagram. What do I actually need in this diagram? I need a nucleus, I need a rough ER, I need the Golgi, and I need a plasma membrane. That's all you need to talk about, okay? Then we need to identify the most likely cellular location of the ribosomes that synthesize CFTR. So the place where we're going to make them is going to be in the rough ER, okay? So notice it is labeled, okay? Pick a picture, pick something in the picture and write it, okay? Never leave anything blank. You should at least try, okay? So it's asking you to tell me where. Pick one of these words. If you're wrong, that sucks. If you're right, hey, you got a point. You didn't even know the answer, okay? So talk about the ER. You could say rough ER, that's in parentheses, so you didn't have to say it, but it is could be something you said. And then identify most likely location of the protein that has a substitution, okay? Well, the job of it is to allow chloride ions across the membrane, so it has to be found in the membrane. Like, again, don't leave anything blank, at least try. The worst that happens is you don't get it right. The best thing, you might get it without knowing anything. Yay! And so do notice that both these questions did say identify, okay? Most questions you could write one word, okay? But sometimes I have seen where they require you to write a sentence. So can you please be safe and just write, the location is the ER. The location is the plasma membrane. It did not take you that long to write a couple extra words. Please, I want you to get all your points possible, because if you know the information, you should at least get your points, okay? So that's all that I have in terms of big idea 4. This is actually like my fastest one so far. So I'm gonna give you a moment that if you want to type any questions you have in the chat, you can type those in the chat. I'm going to talk for a second just about like what resources I have on my website, in case like you're needing some resources. So for some reason, I don't know why, but it closed. So here is my website, apbiopenguins.weebly. Of course, the internet doesn't want to cooperate. So if you go to apbiopenguins.weebly.com, you've got tons of resources on there. Oh my gosh, I just froze and it just blinked. Anyways, I don't know what's happening. Anyway, so API Penguins, there is quizzes, games on there. There's a ton of like all of my videos with Marco Learning are on there. Tons of resources. So anyways, does anybody have any questions in the chat? Like this literally worked right before I started the video. I swear it did. Like, I promise you my video worked. Anyways, okay, so I don't see anyone asking any questions. So if you don't have any questions, then I'm going to remind you to make sure that you're following all of our, media. So you've got Marco Learning on TikTok, you got Marco Learning on Instagram, you got them on YouTube, which you found them because we're right here on YouTube, as well as you've got me on Instagram. I'm posting daily on my Instagram stories, Monday through Friday. Review, we're all the way in Unit 5. If you want to go see the older ones, they're all found on my website. Hopefully you're not blocked like I'm blocked. So hope everyone has a wonderful day, and API Penguins just